Hello. In this video, we will briefly discuss single-cell RNA sequencing and the 10x Genomics platform. We will then talk about the R program Seurat, which we will use to analyze our single-cell RNA sequencing data. This will be an introduction to the work you will be doing on day 8 of the short read workshop. Single-cell RNA sequencing is a method of determining the RNA expression profile for individual cells. This is useful for answering a number of biological questions. For example, single-cell RNA sequencing is used to classify cell types in mixed populations of cells and can uncover rare or previously uncategorized cell types whose signal would otherwise be overwhelmed in a heterogeneous cell population. In recent years, this has become the gold standard for determining different cell types over other methods like cell surface protein expression. Here's an example of a study from 2021 where the authors used single-cell RNA sequencing to categorize the blood cell type populations in both typical and Down syndrome fetal bone marrow. They identified a number of lesser-known blood progenitor cell intermediates and showed a disruption in the erythroid and myeloid cell lineages in Down syndrome bone marrow. These findings are consistent with the blood malignancies we see in children with Down syndrome. Probably the most well-known biotech company in the single-cell RNA sequencing field is 10x Genomics. They use a microfluidics droplet method, which isolates single cells with a barcoded bead in a droplet. This gives you RNA molecules that have cell-specific barcode and a unique molecular identifier, or UMI. These RNA molecules can then be assigned to an individual cell. 10x Genomics offers a set of analysis pipelines called Cell Ranger. We will be using Cell Ranger Count during day 8 of the workshop, which inputs FASTQ files and performs alignment, filtering, barcode counting, and UMI counting. It then outputs a count matrix that we can analyze with the R program Seurat. As a note, Cell Ranger also outputs a loop file, which can be viewed on the Loop browser, which is a desktop application you can use to visualize your single-cell RNA sequencing data. Now, someone else is going to talk about R analysis using the program Seurat.
Hi, so my name is Jesse, and I'm going to talk about what we do with these Cell Ranger outputs. And so, as Chris just mentioned, Cell Ranger is the method of aligning our RNA sequencing reads to the genome and assigning, of calculating the number of reads that each cell has for a given gene. So once we have the outputs from Cell Ranger, we have these gene matrices. We can now load these files off of Cell Ranger into our local computer, into our studio on our local computer, and specifically into this program called Seurat. In Seurat, Seurat is contains a comprehensive set of functions that allow us to normalize and scale and process our data, as well as dimensional reduction of this very high dimensional data set. And then it allows us to cluster and visualize our data, as well as it provides functionality for differential expression, as well as some other additional analyses. And then from this Seurat analysis, we can then go on to do even further analysis using different programs, mostly still in R, where we can look at different pathways that are enriched across different cell types or different variables and perturbations, as well as do things like pseudotime analysis and velocity analysis, which are more advanced analysis for specific types of single-cell sequencing experiments. So Seurat is a program that was developed in California, Satija Lab, and they named it Seurat after a painter, George Seurat, from the 1880s. And this is an example of one of his paintings up here. And he painted, he was an impressionist painting, and he used a series of points. And so it was referred to as pointillist painting. And the authors and the developers of this software named it after George Seurat because they want to invoke this analogy between the intricate spatial patterning of single cells and pointillistic paintings. And so Seurat, as I said, has this comprehensive set of functions that allow us to process our data, assess the quality of the data, normalize it, and run dimensional reduction, and eventually visualize our clusters. Down here, this is actually a plot that I made from single cell sequencing experiments conducted in our lab. And this is from skeletal muscle, and these are similar to the data sets that we'll be analyzing tomorrow in the actual workshop. And you can see that there are different cell types that are being segregated based on their transcriptional profiles. So we have muscle stem cells down here, and we have muscle nuclei. Muscle is a syncytium, so every single cell contains many, many nuclei, and we can see them here. But we also have immune cells and fibroblasts and smooth muscle cells and Schwann cells and other cell types that are present in skeletal muscle. And so what single cell sequencing enables us to do is to identify these different populations and segregate them based on these transcriptional differences. And so Seurat, again, will eventually allow us to get to this point, starting from the outputs that we get from Cell Ranger. So more specifically, there's this workflow in Seurat that we'll be going over tomorrow in our worksheet. And we start by processing the individual Seurat objects. And this includes adding metadata, which is essentially designations that we assign to individual experiments that correlate cells with the experiments that they came from. So we can, if we did experiments in Down syndrome or not Down syndrome, we could add metadata that will identify cells as having come from one of these two experiments, so that when we combine the cells later and we visualize them, we can identify which cells came from which experiments. So in brief, what we do is we add this metadata, we merge these Seurat objects, we normalize the data for differences in sequencing depths across these different experiments. We find genes that are most variable across different experiments, so that way we kind of bring to the foreground genes that are most important, and so we don't have to worry so much about genes that are more uniformly expressed across all different cell types, and we can focus on the gene expression differences that really distinguish different cell types. And then we're going to scale the data, run a linear dimensional reduction first, principal component analysis, and then we're actually going to integrate our objects. So Seurat offers a pipeline for data integration, and this will be important basically for anyone that is doing experiments where they want to compare two different conditions, because we want to ultimately combine our data sets and visualize them together, so we can actually visualize the differences between the data sets and visualize the effect of a given variable on gene expression. And so first, we actually split the Seurat objects before identifying the anchors so anchors are genes that are going to be used, that are expressed somewhat similarly between two conditions, and we can use these to actually integrate these objects. So they're anchors that are helpful for scaling and integrating these two data sets together. And then finally in Seurat, we're able to plot and cluster our single cell sequencing using UMAP, which is as Chris said, which is the predominant form of visualization and dimensional reduction that is used for single cell sequencing these days.